When we're thinking about both breeding of forest trees and deployment of seed or seedlings from tree improvement programs, it's important to think about how those seedlings or seed are related to each other. This both gives us information about whether heritable traits exist in those progeny or the degree of relatedness among those progeny, which sometimes can speak to variation that we might observe in the field. So starting out with, this is a situation that could exist in a wild forest stand or in a seed orchard, for instance, where seed are being produced for later reforestation. In this case, we have a mother tree and several father trees, where these fathers are producing pollen that are pollinating flowers or ovulate strobili on the mother tree. Now, of course, most forest trees are monoecious, which means there are both male and female reproductive structures on those trees. But in the case of most forest tree breeding, we usually track the mother. So for instance, we collect seed or cones or fruits from a mother tree and track that mother. So when, for instance, we have a tree in the forest from which we've collected seed, and that tree has been open pollinated, meaning it was pollinated by the pollen cloud that was floating around from trees in the vicinity, then the offspring derived from seed collected from that mother tree are related to each other as half-sibs. That means we know that, for instance, these two trees share that mother tree in common, but we don't necessarily know who the father was. It may have been the same father, that's certainly possible, but it may not. So those offspring are referred to as half-sib or open pollinated seedlings. And there's a fairly high degree of genetic variability among those trees. Now, certainly, if we collected seed from many different trees in the population, and there were many different mothers pollinated by many different fathers, that would be even less degree of genetic similarity among those offspring. So in this case, all these offspring here share a mother in common, and there's that degree of genetic similarity among them. So a higher degree of genetic similarity exists when we have full sibs. So we have full sibs when we have a mother tree that was pollinated by one father. Okay, so you can see in this case, I've sort of enclosed that mother tree and those female reproductive structures, and pollen has been introduced to pollinate the female reproductive structures on that mother, so that we know then that the offspring from that tree are related to each other as full sibs. So any two trees or any two progeny, so we refer to those offspring as progeny, share a mother and a father in common. Obviously, these half-sib, full-sib relationships can exist in human families as well, in the same way, same type of possible combinations. So obviously, full sibs have a greater genetic similarity to each other than do half-sibs. And there's less genetic variation within a full sib family than there is in a half-sib family. Now, half-sibs and full-sibs are a common relatedness, and you can buy, for instance, seedlings that are either half-sib offspring progeny or full-sib progeny. Full-sib progeny, if you buy them, are more expensive because it's more expensive to produce those. So here's an example of how full-sib progeny are produced. This is Dr. Steve McKeon. He's retired now, but he was longtime director of the tree breeding cooperative at North Carolina State University, and he talks about creating full-sib crosses.
And I'm going to demonstrate today the art and science of controlled pollinations. This is part of our saw timber elite breeding program, and we're making crosses among very highly selected parent trees, some of the very best material that we have in the program. What I want to show you is inside this pollination bag are female strobili. We call them flowers, but they're actually strobili. They are inside this PBS International, which is a woven bag that actually isolates the female strobili and protects them from the outside pollen. Because what we want to do is make a specific cross between this tree, which we have top grafted actually a couple of years ago, and the pollen that I'll be showing you here in a moment. It's a fairly typical pollination day, a bit breezy today, not too bad. And in fact, inside the pollen bag, there has been a bit of wind damage on some of the strobili. That's not unusual for this time of year. They had 30 and 40 mile an hour winds here a couple days ago, so we did have some damage. This is a cyclone pollinator. It was actually a device used by plant pathologists to collect spores. The process has been reversed. Rather than sucking in spores, we're actually going to be delivering pollen in a reverse direction from collecting spores. We're actually delivering the material to the tree. This pollen has been kept in a cooler. I took it out just a few minutes ago. This is one of our selections that I will be making the control cross on. And you can see it's been capped. We want to minimize pollen contamination. We do everything possible to minimize getting pollen from another tree into this. You can see the pollen, I put about a cc of pollen in there. And I'll take off the cork, set it over here. We keep that there just for protection because these needles are sharp. And I'm actually holding the bag securely and poking that needle into the pollination bag. And you'll see here, I'm holding the pollen down and start squeezing the bulb. And you can see what happens. We are just putting a massive amount of pollen, about a milliliter or a cc. And the nice thing about these cyclone pollinators is that after you deliver the pollen, you can squeeze it several times, and I do it 10 or 15 times just to circulate the pollen around in the bag. And it really helps to deliver a lot of pollen to those strobili and will increase seed yield. One of the things we do is we take a piece of tape. This is high quality plastic tape, and we actually cover that hole that I just made because we want to minimize or come as close to minimizing or eliminating any outside pollen contamination. And you can see there's the strobili that have been pollinated. These are about ideal for pollination, stage 4, late stage 4, early stage 5. And we will come back probably in a couple of weeks, maybe 10 days, remove this bag, and then 18 months from now, the cones will be mature and we'll be harvesting this control cross. This is a technique that's been used for years in pine. You can see here in the seed orchard, in our breeding orchard, there's lots of pollination bags. We put up over 300 this year. And the pollination season is just starting, but here over the next week or so, we should have all these crosses made.
So that sort of gives you an idea of what it takes to make a full sib cross. It's a lot of work. You've got to get up into a mature tree, place the bag, come back later, do the pollination, come back later, remove the bag, and then come back later and harvest the cones. Dr. McKeon pointed out that in this orchard there's about 300 bags. I want to point out that that is essentially a research effort going on. Those crosses are being made for the purposes of breeding, and they'll test the offspring from those crosses. When this is done commercially in commercial seed orchards, they will deploy thousands and thousands of bags. So, and then the seed are either sold commercially, or they're used to reforest that entity's forest land. So just gives you an idea of the magnitude of effort required for creating full sib crosses. Clonal reproduction is the final type of reproduction I want to talk about, and this is also called vegetative reproduction. Or asexual reproduction. It just involves one individual. So in this case, there's one, you could call this a mother tree, but there's no father involved. And in this case, there are a number of different ways that this happens, and I'll talk in more detail about it. But essentially, offspring are produced that are all genetically identical to each other. They all have the same genotype as the mother tree. Okay, so these are all the same genotype. So clonal or vegetative reproduction produces offspring that are genetically identical. So clearly, clonal offspring have the least degree of genetic variation compared to full sib and half sib. Obviously, the genetic variation among clones is 0. There is no genetic variation. There's a number of different mechanisms of vegetative reproduction in forest trees, and we've referred to some of these in previous lectures, so I'm not going to dwell on some of these. But for instance, root sprouting is one mechanism of vegetative reproduction. Root sprouts arise from adventitious buds in the roots. And aspen is a really common root sprouting species. This photograph shows someone standing in a little aspen clone or grove that derived from root sprouts, and they've cut the upper part of the trees off and washed the soil away, and you can tell how the root system is all interconnected, and likely all of those trees are genetically identical to each other. We talked about Pando, the large aspen grove that is a single clone of aspen that derives from root sprouts. Stump sprouts, another example. Sometimes we refer to stump sprouts as coppicing. So if a tree is cut down or broken off at the base, dormant and sometimes adventitious buds in the stump will sprout and produce additional trees. So sometimes if you see a ring of trees in the forest, that is often trees derived from stump sprouts. Stump sprouting and generally all forms of vegetative reproduction are more common in angiosperms, but they do occasionally occur in some gymnosperms. Rooting of branches or branch cuttings can occur in some angiosperms. So this is an example in black cottonwood. In black cottonwood, which is common in the western United States, branches will break off. This is a riparian species. Those branches will break off fall into the river, flow downriver, and become lodged in sandbars. And then those branches will actually take root and sprout new plants. So that's an example of a cottonwood branch that's lodged in a sandbar and is sprouting. And you can actually see waves of vegetative reproduction in sandbars, for instance, here. This is a little stand of black cottonwood that's establishing from branch rooting. Now, this is also done commercially. This is hybrid poplar. So these are hybrids of black cottonwood with eastern cottonwood. It's an artificial hybrid, and these hybrids grow remarkably fast. And this is a clonal stand that was reproduced from branch cuttings from a single clone of hybrid cottonwood. And these trees are probably 50 feet tall and are probably 3 years old. So growing very rapidly, a single clone. And this is another view of that same plantation, and you can see the uniformity in this stand. You know, this dark feature here on the stem, it occurs at the same place. The lower branches of these trees all occur at the same place. So these are rooted cuttings stuck in the ground and producing clonal stands with remarkable uniformity and also very rapid growth. Again, some gymnosperms will vegetatively reproduce. So shortleaf pine retains dormant or adventitious buds, and when killed back by fire, for instance, it will resprout from a basal crook at its base and actually vegetatively reproduce after fire. So this is thought to be an adaptation to fire in shortleaf pine. So redwood, Sequoia sempervirens, is another species that can vegetatively reproduce. So here's an example of redwood that's been killed in a stand replacing fire, and they are resprouting from the base. There are dormant or adventitious buds in the base of those trees, and those are resprouting. So that's, again, an adaptation to fire. Now, there also are examples of vegetative reproduction that is artificial. So there's examples in a number of pines where people will take cuttings from desirable genotypes of pine, treat them with hormones, and then root those cuttings and produce clonal offspring in that way. So this has been done in loblolly pine and in radiata pine, but that normally wouldn't happen in nature. There are that can be done artificially. There also are examples of this being done using somatic embryogenesis. So with somatic embryogenesis, it's actually a tissue culture technique where the tissue, somatic tissue from within a seed, is basically multiplied out to produce many copies of an individual genotype. The advantage of that is that embryonic tissue can be stored on liquid nitrogen and brought out many times to produce, in some cases, maybe millions of copies of an individual genotype. So at the present time, when you buy, you can go to vendors and buy, for instance, clonal loblolly pine. Those generally have been reproduced through somatic embryogenesis. And that's just another form of vegetative reproduction. It's artificial, not a natural form of vegetative reproduction, but it's another way of producing commercially clonal offspring from pine. The final example of vegetative reproduction I want to talk about is layering. And I'm showing a picture of black spruce because black spruce is a forest tree species that sometimes reproduces by layering. And layering basically occurs when you have a tree growing in a forest and the lower branches are near the ground and become covered in leaf litter, so dead organic matter. And when that happens, those branches that are covered produce roots and shoots and actually will produce a new tree. Obviously, that's a clone. It's genetically identical to the mother tree, and that is layering. Layering is also done commercially by some nursery growers. They will tie branches to the ground and cover them with moss and keep it moist, and that will produce genetically identical plants. So it's used horticulturally as well, but it occurs naturally in spruce. And you know, one theme that you may have picked up here, but I want to point out if you haven't, is that vegetative reproduction occurs in many situations where reproduction by seed is difficult. So black spruce here often grows on organic soils, on deep organic soils made up of dead vegetation. And those organic soils are not very hospitable for seed. Seed will land on the top of an organic soil and very quickly dry out. Most seed require a mineral soil seed bed to germinate and survive. So that doesn't exist in many black spruce bogs. So it appears that layering in this case has evolved as an adaptive reproductive mechanism for black spruce. In black cottonwood, the examples I gave, seed will be carried off downriver and probably carried to the sea. But when branches fall into the river from black cottonwood, they more easily lodge in sandbars and allow reproduction. Stump sprouting and root sprouting provide mechanisms of reproduction after disturbance, for instance. So a lot of these vegetative reproduction mechanisms are likely evolved to situations where reproduction by seed is not possible or is less likely. So, nature finds a way, evolution finds a way. In this case, vegetative reproduction.